Hi, this is Simon Judd. I'm going to talk about how membranes are integrated with aerobic biological treatment processes. Well, in the previous video, I've talked about how membranes can either be placed downstream of a biological process, in which case they're not integrated with the process biology at all, and they're normally referred to as polishing units in those particular instances, or they can be integrated with the process biology, and it's the latter that I'm going to talk about in this particular video. So if we look at the biological wastewater treatment processes as a whole, they can be categorized generally as either aerobic if they use air to carry out the biochemistry, or anaerobic if they don't. There's also anoxic processes, which I'm going to include in with aerobic processes for the purposes of this introductory discussion, because they're configured pretty much in the same way. So within the aerobic process category, we have suspended growth and fixed film processes, which describe the way in which the biology is actually configured within the process. So for suspended growth processes, there are particles, normally referred to as flocks, which are agitated in a tank which is fed with air bubbles, and the oxygen required for the aerobic process comes from those air bubbles. So that requires a certain amount of energy to pump the air into the tank. For a fixed film process, the biology takes the form of a biofilm which is attached to some kind of medium within the process. So looking at the suspended growth processes overall and specific examples of them, the most established is the conventional or classical activated sludge process. For this particular variant of the suspended growth process, the process biological tank has a downstream sedimentation tank which is used to separate the flocks from the water by sedimentation. So it is a two-stage process with two separate tanks. The sequencing batch reactor is pretty much the same thing, where instead of having two tanks, there's just a single tank where those two processes, the biological treatment and the sedimentation, are carried out in sequence, hence the term sequencing batch reactor. The membrane bioreactor is more like the conventional activated sludge, but instead of having a sedimentation tank for separating the solids, a membrane is used instead, which gives pretty much 100% separation of the solids from the liquid. Moving on to the fixed film process, again the most old and established of these is the trickling filter, which is a very simple design. In this case, a medium is set inside some kind of vessel, and water is trickled over the surface of the medium as a thin film, so that thin film of water is in contact both with the medium and with air, from which the oxygen is taken. The biofilm grows on the surface of the medium, so that's where the biological treatment takes place, so that you have basically a very simple process where there's no air pumping required, because the whole thing is in contact with air. So it's very low in energy, but the process is rather slow and the footprint required therefore quite large. The moving bed bioreactor, or MBBR, contains lumps of medium within a tank which is agitated with air in the same way as the activated sludge process. But in this case, instead of having individual flocks, the biology is attached to media, but that media is being agitated. So you have a rather more intensive process than would be the case for a trickling filter. And because the biology is attached to a medium, the medium is much larger than the flocks, so they're rather more easily retained in the vessel. And then you have the much newer MABR, that's the membrane aerated biofilm reactor, in which the medium is actually the membrane. So the biofilm forms on the surface of the membrane, and that biofilm is fed with air or oxygen from the other side of the membrane to feed the biofilm, and the membrane is then immersed in a tank in the same way as the media is immersed in the tank for an MBBR. So it is a fixed film process, and there's rather more detail of that process again in another video. Straddling both the suspended growth and the fixed film variants is the integrated fixed film activated sludge process, which is effectively the activated sludge process with media added to intensify the process to some extent. So those are just a few examples of suspended growth and fixed film processes. There are quite a few others, but this is just to really emphasize the fact that the MBR sits in the suspended growth group, whereas the MABR is a fixed film process.